All these ones, we have been doing it from our part 3. It's not something that we are hearing for the first time. You know the coagulase test and you know the catalase test. We will have to go through the catalase and coagulase again.
No, sir.
So major disease caused by staph aureus are sepsis, pneumonia, wound infection, folliculitis, necrotizing fasciitis, cardiac skin syndrome, and all whatnot is listed here, so you can get it when you are reading. So, like we have said that the abnormal flora of the nose and skin is the same thing this one is talking about. Frequent colonizer of the skin and nasal mucosa is, in other words, what we have said before. So we don't need to go over it again. Major characteristics of Staphylococcus aureus. You start from their gram stain. You know how it appears in the gram. They are in clusters. Then catalase is positive. Coagulase is positive. They are non-motile. They are non-sporing. They don't have spores, and they are not motile. They don't move. They are not capsulated as well. They are facultative anaerobes. The main disease caused by staph aureus. Once you use this acronym, soft pains, you will get the main disease caused by staph aureus. The first one is skin infection. You know, I say you use these acronyms to get the disease. The second one is osteomyelitis. The third one is food poisoning, that is otherwise known as gastroenteritis. We have the fourth one, toxic choking syndrome. The fifth, pneumonia. The sixth, acute endocarditis. The seventh, infective arthritis. The eighth, necrotizing fasciitis. Nine, sepsis and staphylococcus cardiac skin syndrome. SSSS. Then what are the virulence factors? What are the things that is making it to establish itself and cause disease? We have the surface protein, we have super antigen, we have cytotoxin, we have enzymes such as nuclease, lipase. Then the major virulence factors are the coagulase, the collagenase, the hemoglobin protease, protein, toxic shock syndrome, toxins, enterotoxin, exfoliating, etc. These are the major virulence factors. Then, how do they look on the plates? The staph aureus, this is how it looks on the plate. Look at it. When you culture it, this is how it look like. Then, when you culture on the blood agar, the growth occurs abundantly within 18 to 24 hours. So they can easily grow on our culture medium. Looking yellowish or golden yellow colony with or without beta hemolysis. So you can see them with beta hemolysis. Sometimes they do not show beta hemolysis. The mannitol salt agar is the selective medium commonly used for isolation of staph aureus. Biochemical tests, we have talked about it before, catalase and coagulase. Then, what are how do you identify them? The Staphylococcus aureus. Is catalase positive? Hemolysis, showing beta hemolysis. Coagulase is positive. Then we have, what's it called? The CON that you are seeing is coagulate negative Staphylococcus. This S aureus is coagulase negative, positive. So these are the tests that we use in the lab for its identification. Then Staphylococcus aureus, including methicillin-resistant staph aureus, it's one of the most common causes of healthcare-associated infection. Some strains such as methicillin-resistant staph aureus are highly resistant to multiple antibiotics. Any Staphylococcus aureus, they tag as methicillin resistant, before it can be resistant to methicillin, because methicillin is one of the drug of choice for Staphylococcus aureus. That means that organism must have been resistant to so many organisms. It must have been antibiotics. That is why they said it highly resistant to multiple antibiotics. So when you use methicillin in the laboratory and it's resistant, that means that if you subject it to other drugs, it will also what's it called, show resistance. But there's one agent that has um, shown what's it called, um, shown to be potent against the methicillin-resistant Staphylococcus aureus. The agent is vancomycin, but it will interest you that we still have vancomycin-resistant Staphylococcus aureus. So that means that the organism, as they are getting new agents, they are also devising new way of evading the drugs. Are you there?
Yes, sir.
So we'll go to the less important one. Not that they are less important like that, but which they really cause infection. We have Staphylococcus saprophyticus. They are primarily found in the mucosa of the genital tract of young women. That's where they normally found, and causes urinary tract infection. And then in this group, it is second to E. coli as a cause of community acquired UTI. So what we are saying is that after you have taken Escherichia coli as cause of UTI in the community, the next one is Staphylococcus saprophyticus. Then we have Staphylococcus epidermidis. This one is a normal flora of human skin. You can see saprophyticus is a normal flora of the mucosa of the genital tract. So this one is the normal flora, the epidermidis is normal flora of the skin and mucosal membrane. They are opportunistic pathogen. That means that they are there normally. That is their normal habitat. But when the immune system is compromised, they become pathogen. That's why they refer to them as opportunistic pathogen. They are coagulase negative because it's only Staph aureus that is coagulase positive. That's why they refer them as coagulase negative staphylococci, cons. So when you hear cons, you know the meaning of cons. Then we have streptococcus species. You know what we are discussing under gram-positive cocci of medical importance is the staphylococcus, the staphylococci. Let me just say staphylococci because it's not, there are about 30 species, and we have the streptococcus species which are called streptococci. The streptococci are spherical gram-positive cocci arranged in pairs or chains. You can see the other one is in cluster, this one is in chains. They are catalase negative. Once you do your catalase and it's positive, you know it's Staphylococcus aureus. And when you see it's negative, you are suspecting streptococcus. Traditionally, streptococcus are identified based on hemolysis type shown on blood agar cell wall. And then we have Streptococcus pneumoniae, we have the Friedian streptococca and the alpha, which are alpha hemolytic. The Friedians are alpha hemolytic and the Streptococca pneumoniae. Then Streptococca agalactae and Streptococcus pyogenes are beta hemolytic. So what this one is saying is that Streptococcus species, we have strep pneumo, strep pyogenes, strep agalactae, strep Bifidus streptococcus. But out of these four, that does not mean that there are not more than four, but those are the ones that are very important to us. We know we are dealing with the one of medical importance. And the strep pneumo and viridians are regarded as alpha hemolytic. They show alpha hemolysis, while strep pyogenes and strep agalactae are beta. They show beta hemolysis on blood agar. Then let's go to strep pyogenes. Strep pyogenes, they are regarded as group A streptococcus, also known as flesh-eating bacteria. It's the leading bacterial cause of pharyngitis. They call pharyngitis, which is the throat infection, and they cause cellulitis. It's also called impetigo, necrotizing fasciitis, scarlet fever, and all whatnot. Is everything is listed here. Then we have okay, we have streptococcus. Then what are the characteristics of streptococcus pyogenes? Um, These gram-positive cocci are mostly occur in chains, occasionally in pairs, and then they belong to group A, Streptococcus. Then it is constitutive agents of pharyngitis. We have talked about all this, so don't let us repeat them again. Uh, so this is what we are talking about, the hemolysis on blood agar. Can you see the hemolysis on blood agar? Hello? Hello?
Yes, sir.
You
We
do,
can see, sir.
can't you see the hemolysis? Somebody is saying no. I am pointing at it. Can't you see around the colony? You don't see something like a capsule around the colony. Can we see this blood agar plate?
Yes, sir.
Are you seeing the beta hemolysis?
Yes, sir.
Okay. Somebody is saying no. I hope your network is very fine there.
Clear,
Then
Moses.
you say what? Then the major characteristics of Streptococcus pyogenes: gram-positive cocci, non-motile, non-sporing, fastidious organ, grow well in 5% sheep blood agar, and chocolate. So, catalase negative, bacitracin sensitive. Then the virulence factors of Streptococcus pyogenes. We have hemolysins, we have pyrogenic exotoxins. These are the things that are making it to cause disease, attaches them and cause disease. All these are factors that are the strep pyogenes are using to evade a normal immune system. They are listed 1 to 7. Then disease caused by strep pyogenes, use acronyms NIPUS. You will see, you will get the disease. The first one is necrotizing fasciitis and myositis. The second one is impetigo. The third one is pharyngitis. The third one is pneumonia. The fourth one is lymphangitis, till you get to scarlet fever and cellulitis. Now, non-invasive disease, the strep pyogenes causes both non-invasive and invasive. Then the non-invasive disease is streptococcus sore throat, that strep throat, then impetigo, then the invasive, that's necrotizing fasciitis, streptococcal toxic shock syndrome, STSS, cellulitis, bacteremia, pneumonia, puerperal sepsis. What are the key tests that are used to identify strep pyogenes? We use gram staining, we use culture, we making use of beta hemolysis. Then we use two hemolysin, that's streptolysin O and S. That's why we have ASO, anti-streptolysin O and S. Biochemical tests that we use, catalase is negative, bacitracin sensitivity. So this streptococcus pyogenes is sensitive to bacitracin. If you put bacitracin disc on a blood agar or chocolate agar that is suspected to be streptococcus pyogenes, you will see a clear zone of inhibition. Then we have all these tests, they are either positive or negative. like I was saying, the anti-streptomycin whole so test this titer is not done for diagnosis of streptococcus aureus, but for sequela, and these are tests that when bacteriology was bacteriology that time in the UCH, all these tests were often done. But I'm sure that most of these tests, I don't think they are doing it again. Now, the next one that is very important to us is Streptococcus pneumoniae. So, Streptococcus pneumoniae is a normal upper respiratory tract flora and most commonly cause of community-acquired pneumonia. It causes bacteremia, meningitis, otitis media. These are invasive diseases. When you have bacteremia or meningitis, it's not a disease that you joke with. Conjunctivitis, and they are gram-positive diplococci. They produce alpha hemolysis on blood organ, while the strep pyogenes produces beta hemolysis. Strep pneumo produces alpha hemolysis. While the other one is bacitracin sensitive, this one is optochin sensitive. It's another disc that they use to confirm the presence of strep pneumo. What are the properties of strep pneumo? Normal flora of nasopharyngeal and oropharyngeal flora. It's an important etiological agent of upper and lower respiratory tract infection. Bacteremia, septicemia, all whatnot. Streptococcus pneumo is gram-negative cocci, and they occur singly in pairs, that is diplococci. It's either they occur singly or they occur in pairs, which is diplococci. They are non-motile and they are encapsulated. Non-motile and encapsulated. Strep pneumo is fastidious bacterium that grow in the presence of 5% carbon dioxide or in a candle jar. If you don't want to miss strep pneumo, just incubate the organism in candle jar. And that's why most of the time, at the time we were in UCH, the candle jar were always in use because we don't want to miss strep pneumoniae. Then we have this is gram staining of strep pneumo. What are the diseases caused by strep pneumonia? They cause bacterial pneumonia, the first one, the meningitis, the second one, otitis media, common in children, and sinusitis, common in children, bronchitis, bacteremia. These are the diseases they cause. Then their own virulence factor, these are listed. Then we have autolysin as one of the major virulence factors. We have pneumolysins, polysaccharide capsule. I told you the other time that strep pneumo is encapsulated. It has capsule. So, laboratory diagnosis, these are the colony it's based on the laboratory diagnosis, based on the characteristic shape of the organism. We know the characteristic shape, diplococci, gram-positive diplococci. There are biochemical reactions susceptible to certain diagnosis disc. We talk about optochin the other time, sensitive. The samples that you can recover strep pneumonia for is mostly respiratory specimens, sputum, lung aspirate, pleural fluid, body fluid, e.g., blood, cerebrospinal fluid. You can still get strep pneumo from them. Strep pneumo, if in a suspected sample, must be transported promptly to the laboratory within 1 to 2 hours. If not, you won't be able to get them, they will just die off. So you need to. So what are the culture? How do you culture them and identify? You can culture them on blood agar. You use alpha hemolysis for them. This is strep pneumo is alpha hemolysis. They are optochim sensitive. So these are the things that you can use to identify them in that flow chart. Are we there? Hello, are we there?
Sir.
Yeah.
Yes, sir.
Yes, sir.
Yes, sir.
Then you can still perform gram staining on the sample. Okay, great. You can perform gram staining on the sample. This is how it look like. They are gram positive diplococca. So can't miss them. Culture and sensitivity, you can culture them on blood agar and chocolate agar, especially enriching 5 to 10% carbon dioxide for 24 hours or more. We can incubate them for 48 hours. If they are not coming up the way you wanted, you can further incubate for another 24 hours. So these are the colonial morphology, how they look like on the colony. So
That's greenish hemolysis.
that's alpha, yeah. You can only get the greenish on chocolate. Do you get? On chocolate agar. That's where the greenish will be very pronounced. Are you getting it? But the greenish on the blood agar is not that pronounced. But if you use chocolate, then you can see that greenish color very well. Then these are the biochemical tests for strep pneumo. Catalase negative, optochin sensitive, biosolubility test is soluble, hemolysin is alpha hemolysis, oxidase test is negative, and then motility is non-motile. So this is optochin we are talking about. You can see it's sensitive, see that clear zone, zone of inhibition. So then agalactiae, it's not something that we are going to concentrate upon. You can read that one. There's no much under it. It's just it's also a group B streptococcus, that is the colonized genital tract of some women, leading cause of neonatal sepsis, pneumonia, and meningitis. Then we have Viridian streptococcus, which are alpha hemolytic streptococcus. But unlike strep pneumonia, they are toxin resistant. You can see, both strep pneumo and Viridian streptococcus, they produce alpha hemolysis, but you can distinguish the two. You can distinguish the two. While strep pneumo is optocin sensitive, Viridian streptococca is optocin resistant. Then we have group D streptococca. This includes enterococca, which is e.g. Enterococcus faecalis and Enterococcus faecium. So we have micrococcus, we have peptococcus, and we have peptostreptococcus. These are less important. You can read up. I just put 3 or 4 lines, maximum 5 lines for them so that you have idea. Then we'll go to Neisseria. Neisseria is not gram-positive cocci, it's gram-negative cocci. Do you get what I'm trying to say?
Intracellular.
You say yes. So I just put them all because there is what's it called? is cocci. That's why I just put them together. The one that we have treated is gram-positive cocci. This one is gram-negative cocci and gram-negative diplococci. You know, Streps is gram-positive diplococci. I mean, Strep pneumo is gram-positive diplococci. While Neisseria is gram-negative diplococci. Do you get the difference between the two? The Strep pneumo, the two of them are diplococci, but one is gram-positive, the other one is
That one
gram-negative.
is gram-negative, yes.
That's the difference. But the two of them are diplococci.
Yes.
We have two major genus under the Neisseria, although there are many of them, but the one that are very important to us is the Neisseria meningitis and Neisseria gonorrhoeae, and then which are strict human pathogens responsible for meningitis and gonorrhea, respectively.
Gonorrhea.
So, the genus Neisseria contain large organisms that cause human disease. You know, I told you there are many, but the one that we are going to concentrate upon is the Neisseria meningitis and Neisseria gonorrhea. So these organisms are gram-negative diplococci, the way I told you the other time. They are gram-negative. So don't mix it up with Strep pneumo. Strep pneumo is gram-positive diplococci, while Neisseria is gram-negative diplococci. So the genus Neisseria belongs to the family Neisseriaceae, and they are gram-negative diplococci. They range 0.6 to 1.5 micrometer in size. They are non-motile and do not produce endospores. So they are aerobic with normal growth occurring between 35 to 37 degrees Celsius in the presence of humidity and 5% carbon dioxide. You can see these ones also require 5% carbon dioxide. That means that when you do candle jar incubation for Neisseria gonorrhea and Neisseria meningitis, you can recover them, just like for strep pneumoniae. So, and they have another characteristic. They can survive in an anaerobic environment. Though they are not anaerobic organisms, but they can survive there. So some species may express a capsule. So some may be encapsulated while others may not be encapsulated. Then microbiology and laboratory diagnosis, like Neisseria gram-negative, oxidase positive. You can see the strep pneumo is oxidase negative. This one is oxidase. The Neisseria, they are oxidase positive. They are also catalase positive, except this non-important one, Neisseria bacilliformis and Neisseria elongata. So now let's go to gonococcal infection. The genus Neisseria was originally named after the German physician A. Neisser Murray. So two members of the species, Neisseria gonorrhoeae and Neisseria meningitis, cause serious human diseases. We have talked about this before. So while Neisseria gonorrhoeae is spread primarily through sexual contact, the meningitis, the Neisseria meningitis is a disease of the meninges. You know, meninges now is brain cells are called meninges. So that's why Neisseria meningitis affect the central nervous system. It causes a disease called in Yoruba yirun. So it attacks the central nervous system, unlike gonorrhea that goes to that attacks the genitalia. But both male and female genitalia, that's where that one is causing its havoc. So gonorrhea. Neisseria gonorrhea is non-spore forming gram-negative coccus that characteristically grows in diplococca, in pairs rather, and what is pair is diplococca. So adjacent side flattened. It does not have flagella. That means that it's not motile. Can move based on extension and retraction. The process is not twitching. The movement in this area is called twitching because it's not using flagella to
Or the
move.
pillar.
It's using pillar, yes, type 4 pillars to move. And gonococcus closely resemble the meningitidis. You know, the two of them, they are like twins. So the only thing is that the habitat that the two of them are affecting are different, but they are like twins. So hardly can you differentiate the two of them. The only thing that you can use to differentiate them is from which sample did you get them from? Is it from a genital sample or from a cerebrospinal fluid? Are we together? Are we there?
Yes, sir.
Okay, the meningococcus. This one is caused by Neisseria meningitidis, the meningococcal disease from the meningococcal disease. It's caused by Neisseria meningitidis. They are non-sporing, oxidase positive, non-motile, gram-negative cocci, usually kidney shaped diplococci. And see when you see them, they are kidney shape, just like ewa. So they form like beans. So, and then that's how to identify them under the microscope. What is other? They grow rapidly on blood, chocolate, gonococca, or enriched molar inting agar. It also requires 3 to 10 carbon dioxide environment. So using candle jar, you can also recover Neisseria meningitidis. You can still use modified Martin's medium for isolation of Neisseria meningitidis. Then the characteristics of meningococci, they are complex. They have surface components which include the capsular polysaccharide, the fibril, the pili, lipo-oligosaccharide, which is called LOS, and outer membrane proteins, which is OMP. These are the things that form what I can tell you virulence factor, because these are the things that enable this Neisseria meningitidis to evade immune system of the body. Then the Neisseria meningitidis, we have 13 serogroups. We have A, B, C, D, E, H, I, K, L, S, Y, Z, and W135. You count them, you will see that it's 13. Are you getting it? These are the things that I can bring out in objective. Are you getting it? So don't play with all these serotypes. So know them specifically, because I can try to test your knowledge whether you know the 13 serogroups, and then I can just bring them out. And if you did not read the book, you will miss them out because you will not know the sequence at which they follow one another. So, but the majority of the meningococcal disease is caused by organisms in serogroups A, B, C, and Y. Although the serogroups W135 is also the proportion at which it causes disease is increasing in some area. So know all of them and know the one that are causing disease more than the other. This is a convenient point for us to stop today. We will still have to meet tomorrow, and we'll meet on Sunday, so that let's see how far we can go before other lecturers will wake up and they want to compete with the time that I am using. So I want us to go as fast as we can. The gram-positive organism, I think we have to stop here because we can't treat all of them. Maybe in the next class we are going to gram-negative organism. So tomorrow you have enough to read under gram-positive organisms. So let us just leave it there. So that let us go to gram-negative organisms.
Neisseria, gram-positive or gram-negative, sir?
You say what?
I said the Neisseria, is it gram-positive or gram-negative?
I have told you the other time now. The Neisseria is gram-negative cocci. Why I put them here is because they are cocci.
It's because of what you said. You said we're going to start with gram-negative, what do you call it?
No, what I am saying, tomorrow, gram-negative bacilli. GMB.
Bacilli is okay.
Gram-negative bacilli. This one is gram-negative, but because it's cocci, that's why I put it under this. Are you getting what I'm trying
because
to
we did. Did you treat the bacilli to the gram-positive?
I didn't treat what?
The bacilli that are gram-positive.
The bacilli that are gram-positive. That's what I'm saying. I say we cannot treat all these organisms. This little one, do you know how many pages are that you are going to read?
It's okay, sir. It's okay,
and
sir.
If you want me to give you gram-positive bacilli,
no, sir.
I will give you gram-positive bacilli. But you have enough to read.
Sir, it's okay, sir.
You have enough to read before you read Staphylococcus, Streptococcus pyogenes, Streptococcus pneumo. Then you come to Neisseria meningitidis, Neisseria gonorrhea. You don't try now. Before you read all these things, you don't tire yourself.
No, we appreciate it, sir. Thank you, sir.
Don't tire. Before you go read all these things for exam, mind you, it's not only bacteriology you are doing for exam, or you do parasitology, you do mycology, you do public something. So you know how it is now. It is not only this course that you are reading. In fact, when I am putting up the note, I was even saying that is it not too much for us to read? But since
thank you, sir.
At the end of,
Thank you, sir.
This one is okay, sir. It's okay like this, sir.
Okay, sir.
If you want the gram-positive bacilli, I can give you.
No. We are okay this way.
So I just felt that what we need to concentrate upon is the one that is of medical importance, the one that are commonly encountered, the one that we normally play with, even when we were doing part 3, part 4. Nobody will tell me that it's just hearing Neisseria, whether it's meningitis or gonorrhea today. Nobody will tell me that Staphylococcus is so new to him. Nobody will tell me Strep pyogenes is hearing it for the first time. Nobody will tell me that Strep pneumo is just hearing it for the first time. So we believe that if we set exam on this one, even if they come from NUC, they will know that we have sent a standard question for you. And expect something. Let me just tell you before I forget. Expect something in your exam. Don't expect direct question. It's going to be applied question. Are you getting it? What do you mean by applied question? I can just write a story concerning somebody that was admitted, and it was discovered that he has so. So now you go decode them that I am talking about Neisseria meningitis or Neisseria gonorrhea. Now you go decode whether the staff looks or Strep pneumo I was talking about, Strep pyogenes. Do you get?
Thank you, sir.
So that's what I mean by applied question. I will not go and say discuss strep pyogenes, but I can use the disease they cause to ask the question. Then you will know that I am talking about strep pyogenes. Are you there?
Yes, sir.
This is 500 level.
Yes, sir.
It's not 400 level, so don't expect direct question like that. Even when examiner come, they will say, you cannot. You just write direct question as if you are asking 300 level or 400 students. So I know before the exam we will still do some revision, but let's get to know what we need to know, because after 500 level is graduation. You don't learn any other thing at this level again. So the little that we are able to package, let's know it, and let there be value for the lecture. Any question? Any question?
No, sir.
No, sir.
Okay. Do you enjoy the class? Or I was too fast?
No.
Not really.
Very likely.
Okay. I just felt that I shouldn't be coming to class and be dictating notes for you.
Thank you, sir.
Let's just explain it, and then anytime you pick up the note, then you understand it.
All
So,
right, sir.
and that's why my lecture hour is always between 45 minutes and 1 hour now. So,
Okay.
so that let's try to explain to ourselves. And when you are reading it, you will discover that everything the way we have said it is the way you are reading it. So, tomorrow is another day. God willing, I will come and discuss gram-negative bacilli with us. We have very important one there. We have the Escherichia coli, we have Klebsiella, we have Haemophilus, we have Pseudomonas. Very important ones. We'll discuss them. If we can finish gram-negative bacilli tomorrow, fine. We can't finish it, we spill it over till Sunday. And if we are able to finish, we'll take another topic on Sunday. And then that means that we are doing well. By the time other lecturer will wake up, we are through with bacteriology, MMB 501. Thank you so much, and thank you for having me this night. And thank you for coming to class very punctual, because I want us to start early so that we can finish early. Because it's a night lecture, I don't want you guys to be sleeping in the class. That's why I want us to start early, 8 for 8, so that we can finish before 9. Thank you so much. And tomorrow is another day.
Thank you very much. God bless you, sir.
Good night, sir.
Good night.
Good night.
Good night.
Have a nice day.
Very well,
Thank you.
Thank you.
Thank you. Ẹ̀yin àǹtí mi,
night, sir.
ẹ̀yin àǹtí
Yes,
mi
sir.
àti bọ̀dá mi, thank you.
Ẹ ṣé gan, sir. Ajíláyọ̀, sir.
Àmì o, àmì Olúwa ló ṣe o.
Good night, everyone.
Good night.